Good evening once again. Today's discussion is on DNA transcription. So to get started, maybe we can define what DNA transcription. We'll get started. DNA transcription, if you remember from our previous discussion, we had outlined the central dogma of molecular biology, where DNA is converted to RNA. Then RNA is used to make proteins. So the process by which RNA is made from DNA, that's our topic for today's discussion, which is known as DNA transcription. So what is DNA transcription? As I mentioned, it's the synthesis of a RNA strand. This RNA strand can either be ribosomal RNA, tRNA, or messenger RNA from a template DNA strand. We had talked about the three types of RNA in the previous discussion. The synthesis of these RNA strands from a DNA template strand is what is known as DNA transcription. So to get started, if we remember, DNA is double-stranded, and it being double-stranded, it will have two strands. One will be running in the 3' to 5' direction, whereas the other one will be running anti-parallel in the 5' to 3' direction. So which strand is synthesized by, which strand is transcribed by, which strand is used to make RNA? This brings us to the next slide, where we talk about the two DNA strands. You have the sense or coding strand, and you also have the antisense or template strand. So in the synthesis of RNA, the strand that will be used to synthesize the RNA is known as the template strand. So amongst the two strands, you have one strand that will be running 5' to 3', and then you have one strand which will be running 3' to 5'. So the strand that will be running 3' to 5' is known as the, okay. So I was saying the two strands of DNA, one will be known as the sense or coding strand, whereas the other one will be the antisense or template strand. We remember that one will be running in the 5' to 3', whereas the other one will be running anti-parallel 3' to 5'. The strand used for the synthesis of RNA, known as the template strand, will be the one that will run from 3' to 5'. And the RNA that will be synthesized using that strand will be anti-parallel to that strand, meaning the RNA strand synthesized will run from 5' to 3'. This brings us to the next slide. It's transcription now. We've started on transcription. DNA transcription can be divided into three stages: initiation, elongation, and termination. So in initiation, on the template strand, the double helix of the DNA, on the template strand, if you can see the diagram, on the template strand, you have a sequence. This a sequence known as a promoter. So RNA polymerase, which is the enzyme which will be synthesizing this RNA strand, will read this promoter sequence, and then it will bind. That's in the initiation process. It will bind. Once it's bound, it will begin to unwind the DNA, forming what is known as a transcription bubble. Once it unwinds, the RNA polymerase will then move along the template strand, synthesizing a RNA molecule, a RNA strand, synthesizing all the way until it will reach another sequence known as a stop sequence. Once it reaches the stop sequence, the RNA polymerase will disassociate from the template strand and release the synthesized RNA. So those are the three stages of transcription. You have the initiation, elongation, and termination. Now we talk about transcription in prokaryotes, and then later on we'll talk about transcription in eukaryotes. The basic stages will be the same in both. You have initiation, elongation, and then you have termination. But there'll be slight differences between the way transcription is carried out in prokaryotes and the way it's carried out in eukaryotes. So look at transcription in prokaryotes. In prokaryotes, transcription will be carried out by an enzyme, RNA polymerase, but this RNA polymerase is known as RNA holoenzyme, meaning it's just made up of many parts. This diagram here is showing the RNA holoenzyme, which will be the holoenzyme will have it will have two subunits. So you have one subunit known as the sigma subunit, and then you have this subunit here, which will be known as the coenzyme, which will also be made up of different parts. Amongst the parts of the coenzyme, you have the alpha parts, then you have the beta part, then you have the beta prime, and you also have the omega part. So these also have varied functions. The alpha parts, these are simply needed in order to put this whole enzyme together. Then you have the beta part. This beta part is the one which will have the ribonucleotides, which will be used to synthesize the RNA molecule. And then you have the beta prime part, which binds to the DNA template strand. And you have the omega part there. Not really sure on the function of the omega part, but you can look it up. Then on the subunits, you have the sigma subunit. The sigma subunit is what will bind to the promoter sequence. So, as mentioned earlier, the promoter sequence is just a sequence on the template strand where the RNA polymerase will go to attach in order to start the transcription process. So, in prokaryotes, you have this RNA holoenzyme which contains these different parts. So, you have the sigma subunit of the RNA holoenzyme bind to the promoter region, and then once it binds, the coenzyme will dissociate from the holoenzyme. This is what I was explaining in this slide. So in prokaryotes, you have two promoter sequences. One will be at the minus 35 region, and the other one will be at minus 10 region. So what this simply means is that you have this point here in the diagram where transcription will start. But before this start site, 10 base pairs upstream, you have a promoter region. This promoter region will it's a consensus sequence in that in most prokaryotes, this sequence has been found to contain the same arrangement of base pairs. So you have the TAAT, which is at the minus 10. And then at the minus 35, you have the TTGA, CJ. So this is the region that will be bound by the sigma subunit. And once bound, the RNA holoenzyme, the coenzyme part of the RNA holoenzyme would dissociate and begin the elongation process, which brings us to the next slide. The elongation process. So the coenzyme has separated from the sigma unit, and it now starts to move along the DNA template strand. So it's moving along the DNA template strand, synthesizing the RNA molecule using those separate parts from the coenzyme. You have one part that's binding the template strand, and then you have the other part which contains the ribonucleotides. So, and then you have a part that should be, I believe, the omega part, which will be doing the unwinding. So as the RNA polymerase is moving, you have those parts of the coenzyme working to unwind the DNA strand and then also synthesize a RNA strand. So you have that strand being synthesized, which brings us now to the termination step. So as the RNA polymerase continues to move along the DNA template strand, it will reach a stop point. Once it reaches that stop point, in prokaryotes, there are two ways in which it can end this transcription process. So you have what is known as the row-dependent termination, and you have what is known as the row-independent termination. So in the row-dependent termination, as the RNA polymerase is moving along the DNA template strand, you have a protein known as the Rho factor that will bind to the RNA being synthesized. The RNA being synthesized will have a sequence on it where the Rho protein, Rho protein or Rho factor will bind. And once bound, it will continue moving behind the RNA polymerase. So this RNA polymerase, once it reaches the stop sequence, which is usually a GC region, once it reaches that sequence, it slows down. And when it slows down, the Rho factor will catch up to the RNA polymerase and interact with it. And this causes the RNA polymerase to dissociate from the template strand and release the RNA strand that has been synthesized. In the row independent, once the RNA polymerase reaches the stop sequence, it will then add alternating GC bases. So you have GC, GC, GC. So you have G, CC, something in those lines. And so because those sequences it's added complement each other, you have them base pair to each other. And when they base pair to each other, what they'll form a loop, such as the one shown here. So they'll form a loop, such as the one shown here. Once they form that loop, the RNA strand that has been synthesized becomes stable, and enzymes come in and they facilitate the dissociation of the RNA polymerase. This will bring us now to transcription in eukaryotes. So in eukaryotes, transcription is a bit more complicated than in prokaryotes. You have it undergo the same basic steps where you have initiation, elongation, and then you also have termination, but there'll be a few differences at those steps. Uh, in the initiation, you have what are known as transcription factors coming. So transcription factors are just proteins that will interact with the promoter sequence in order to initiate transcription in prokaryotes. So these transcription factors just so you have this diagram here. Just just a minute. So if you're able to see my screen, you have this diagram here. So you have the promoters in the eukaryotes. These promoters, you have three of them. You have the tata box, the cat box, and the GC box. So the tata box will usually be about 50 within 50 base pairs of the start point. And, and then you have the cat box and the GC box a little further upstream. So in eukaryotes, in order to initiate transcription, you have transcription factors that will come and interact with the promoter sequences. And then once they do that, they will recruit the RNA polymerase. The RNA polymerase will then move downstream to the start point and start unwinding, forming that transcription bubble we talked about earlier on. Once this happens, uh, the elongation process starts. The elongation process is the same as in prokaryotes in that you have the RNA polymerase. Uh, you have the RNA polymerase synthesizing ribonucleotides in the 5 prime to 3 prime direction until it reaches a stop point. So you have the RNA polymerase moving along the DNA template strand, synthesizing the ribonucleotide. In eukaryotes, you have three RNA polymerases, which will be responsible for synthesis of different. So, as I was saying, in eukaryotes, you have three RNA polymerases, whereas in prokaryotes, you only had the single RNA polymerase, which was the RNA holoenzyme, doing all the transcription. In eukaryotes, you have three. So you have RNA polymerase 1, which will synthesize ribosomal RNA. Then you have RNA polymerase 2, which will synthesize messenger RNA. And then you have RNA polymerase 3, which will synthesize transfer RNA. So as mentioned, the elongation process is the same as in prokaryotes. The RNA polymerase will just move along the DNA template strand, synthesizing the appropriate RNA strand for that RNA polymerase. The termination will be different from the prokaryotic termination in that in eukaryotes, termination will end when when the RNA polymerase encounters what is known as a poly A signal. So you have a sequence along the template strand. This sequence will be AAU triple A. So this sequence is the termination signal, the poly A signal. So once the RNA polymerase reaches this signal, it dissociates from the template strand and then releases the synthesized RNA. In the case of mRNA, the synthesized uh, mRNA is not yet ready for uh, translation. It's not yet ready to be used to make proteins. So as a result, this mRNA will undergo what are known as post-transcription modifications or pre-translation modifications. So these modifications, there are just three of them. You have 5' capping, then you have polyandination, and then you have splicing. So in the 5' capping, you have three enzymes that will add a guanyl, a methylated guanyl molecule at the 5' end of the synthesized mRNA molecule. So you have, let me use this so that. So at this 5' end, you have a triphosphate at this 5' end. So in mRNA post-transcription modifications, you have an enzyme, RNA triphosphatase. It will come and it will remove two phosphate groups from the 5' end. Once that is done, another enzyme known as guanyl transferase will come, and to this 5' end, it will add the guanosine monophosphate group, and then you have another enzyme come in and add the methyl group to the guanosine monophosphate group. And in that way, you have what is known as a 5' cap at the end here, which will be a methylated guanosine monophosphate molecule at that 5' end. And then one other post-transcription modification that is done to mRNA in eukaryotes will be polyandination, where you have an enzyme which is known as poly-A polymerase. So poly-A polymerase will come and it will read the poly-A signal, the AAUAAA signal there at the end of the stop signal. So you have poly-A polymerase coming and it will read this termination signal. And so once it reads the termination signal, it will add on to the termination signal. It will continue adding on base pairs. It can add up to about 5 base pairs or more. And then apart from the 5' capping and the polyandination, you have splicing, which is also part of the post-transcriptional modifications. So in splicing, what you have to know is that on the mRNA strand which is synthesized, you have base pairs which code for proteins, and you have base pairs which don't code for proteins. So the base pairs which code for proteins will be known as exons, and those that don't code for proteins will be known as introns. So these base pairs, all of them will be transcribed, but not all of them will be useful for the translation process. So as a result, you have enzymes known as spliceosomes. These spliceosomes are just they are complexes of some other proteins known as small nuclear ribonucleoproteins, which will combine with other proteins and they'll make this spliceosome. So the spliceosomes will come in, and what they'll simply do is that they'll remove the regions which are not coding for proteins. So those are the introns. They'll remove the introns, and then a ligase will come in And what the ligase will do is that it will join the exons. So once the exons are joined, that's what is known as splicing. So the exons are spliced together, they're put together. And those exons, once they're spliced together, what they form is known as the open reading frame. So you have this open reading frame, which will be a region on the mRNA, which will contain exons that code for proteins. So at the end of the post-transcription modification, you have the 5' cap at the 5' end of the mRNA, and you have the poly A tail at the 3' end. And then in between, you have the ORF, the open reading frame. But before the open reading frame, so let's say you had, let me use this very sequence here. So you have this mRNA sequence. Then you have a region which will contain exons and introns. So before you encounter the first exon, you have a region which also doesn't code for proteins. You have one such region at the 5' end, and then you have this a similar region there at the stop signal. The stop signal also doesn't code for proteins, so it will also be an untranscribed region. So at the end of the post-transcription modifications, what you have is you have a mRNA molecule, which will have a 5' cap, a poly A tail, and then you have a 5' and untranscribed region. Then you have the open reading frame. Then you have the 3' and transcribed region, and then you have the poly A tail. So we've now talked about transcription in prokaryotes and in eukaryotes, and this table here just summarizes the differences between the two. So amongst the differences, you have the fact that whilst in prokaryotes, you have transcription and translation, which is protein synthesis, occurring at the same time. Whereas in eukaryotes, you have transcription occur on its own in the nucleus, and then the mRNA will go into the cytoplasm where the translation will take place. And the second point says bacterial mRNA begins. I've already mentioned on that. Okay. The other difference in prokaryotes is that, as mentioned, in prokaryotes, you won't have mRNA post-transcription modifications, whereas in eukaryotes, you have those post-transcription modifications. The other difference will be on the promoter sites. In the prokaryotes, you have the -10 region, which is also known as the primnal box, and the -35 region. Whereas in the eukaryotes, you have the Tata box, then you have the cat box, you also have the GC box. And then the final difference there will be that in prokaryotes, the synthesis of RNA will be by a single RNA polymerase, which is the RNA holoenzyme, whereas in eukaryotes, you have 3 RNA polymerases transcribing the RNA, which will be RNA polymerase 1, 2, and 3. So that will bring us to the end of our discussion. But before we end, we can look at the revision questions. So the first question there says, what is DNA transcription? We covered that in the first slide where we simply said it's a synthesis of a RNA strand, which can either be tRNA, RNA, or mRNA from a DNA template strand. Then the next one says, what is a promoter sequence? We answered that. Uh, a promoter sequence is just simply a sequence on the template strand where the RNA polymerase will bind. And then apart from the promoter sequence, when talking about transcription in eukaryotes, we talked about transcription factors, which will interact with the promoter sequence. But apart from the promoter sequence, you have other sequences known as enhancer sequences, where if the transcription factor binds to the enhancer sequence, what you have is that there'll be an increase in the rate of transcription. And then you also have what are known as silencer sequences. So you have a transcription factor bind to a silencer sequence, and such transcription factors will be known as repressors. So once it binds to a silencer sequence, what you have is that the rate of transcription will be depressed. The third question says differentiate between the coding and the template strand. So the coding strand, which will be the DNA strand, which will be running in the 5' to 3' direction. And the coding strand will also run in the same sequence as the RNA strand, which has been synthesized. So basically, the coding sequence and the RNA strand will have the same sequence. The only difference is that in the DNA molecule, you have thymine, whereas in the RNA molecule, you have uracil. Uh, and then the template strand will be the strand from which the RNA molecule is synthesized from. And that strand will run in the 3' to 5' direction. So the RNA strand that will be synthesized will be from 5' to 3'. That answers on the that answers the third question, which was stated differentiate between coding and template strands of DNA. The fourth question says what are introns and what are exons? So we define these when we're talking about splicing, which is part of post transcription mRNA modification. So you have intros, which are sequences that don't code for proteins, and then you have the exons, which are the sequences which code for proteins. Then the fifth question says describe the processes involved in mRNA pre-translational modification. So we talked about those. Those are the 5' capping, those are the polyandrylation, and the splicing. So if there are no questions, that will bring us to the end of our discussion. Thank you very much for attending. Hope to see you in the next session.